Hello and welcome to my academy. Today we are going to discuss about the electron transport system or electron transport chain. Okay, let's get started. When the inner mitochondrial membrane is disrupted by a detergent, the various electron carriers can be isolated as part of four distinct asymmetric membrane spanning complexes identified as complex 1, complex 2nd, complex 3rd, and complex 4th. Complex first is also called NADH dehydrogenase. It contains one molecule of flavin mononucleotide and 6 to 7 iron sulfur clusters. Complex second, also called as succinate coenzyme Q reductase. Succinate dehydrogenase, an integral mitochondrial membrane bound enzyme, is an integral component of complex second. It converts succinate to the fumarate during Krebs cycle. Complex third, also called cytochrome B7 complex. The complex consists of cytochrome C1, iron sulfur, and cytochrome B. Complex 4 is also called as cytochrome C oxidase. It consists of Hem A, Hem A3, Cu A positive, and Cu B positive. The reduced coenzymes, that's NADH and FADH2, are released during the glycolysis and the Krebs cycle. The electrons are transferred from NADH and FADH2 to oxygen, forming water, through a series of electron carriers present on the inner mitochondrial membrane. In the matrix of the mitochondria, high concentration of the proton is there. The protons must be transported into the intermembrane space. This creates an electrochemical gradient. The electrochemical gradient exerts proton motive force to generate ATP. First of all, we will discuss how the electrons from the NADH pass sequentially from one complex to the another until they are finally transferred to the oxygen, which has the highest affinity for electrons. When NADH is electron donor, electrons enter the respiratory chain by means of complex first. NADH is oxidized into NAD plus H positive plus 2 electrons in presence of an enzyme called NADH dehydrogenase. The electrons are accepted by the flavin mononucleotide and is thus reduced, which sends these electrons to the iron sulfur clusters. And ultimately, these electrons are received by ubiquinone molecule, which is further reduced into ubiquinol. The energy released by the electrons is used to drive the four protons from matrix into the intermembrane space. Ubiquinol loses two electrons and is oxidized back into ubiquinone. Again, the energy released by the electrons is used to drive the protons from matrix into the intermembrane space. Here are some facts about the ubiquinones. Ubiquinones are hydrophobic and uncharged, and hence can migrate along the hydrophobic core of the membrane. Now, here these two electrons are again accepted by the iron sulfur clusters. Thus, two more protons are pumped into the intermembrane space. Now we have total of four protons that got pumped at complex third site. The electrons are now transferred from iron sulfur cluster to the cytochrome C1. In between the complex third and fourth, we have a mobile electron carrier called cytochrome C. This cytochrome C carries single electron at a time from complex third to complex fourth. These electrons are first accepted by the copper atom of the complex fourth and are then subsequently transferred to the other cytochromes until they are finally accepted by the final acceptor, that's oxygen, forming water. During this process, two more protons are pumped into the intermembrane space from the matrix. This completes our electron transport chain for the NADH electrons. Thus, in total, 10 protons are pumped into the intermembrane space by NADH electrons. 4 protons at complex first, 4 protons at complex third, and finally 2 protons at complex fourth. Thus, for every NADH molecule, a total of 10 protons are pumped from matrix into the intermembrane space through the complexes of electron transport chain. When FADH2 is the electron donor, the complex one, that's NADH dehydrogenase, do not participate in the electron transfer process. Rather, complex second, that succinate coenzyme Q reductase, is recruited to take part in the electron transport process. It should be noted here that succinate dehydrogenase, an inner mitochondrial membrane bound enzyme, is an integral component of complex second. It converts succinate to fumarate during Krebs cycle. The electrons released from the conversion of succinate to the fumarate are accepted by the FAD and is thus converted into FADH2. The electrons from the FADH2 are received by the iron sulfur cluster. Finally, these electrons are received by the ubiquinone. Thus, ubiquinone is reduced into ubiquinol. The energy released from the electrons is used to drive the protons from matrix into the intermembrane space. Thus, total of 4 protons are pumped into the intermembrane space from the matrix. Now, from here, the story is almost the same as in case of the NADH electrons, except that complex first has no role to play in case of FADH2 electrons. In the similar fashion as in case of NADH electrons, 2 more protons are pumped at the complex 4 site. Thus, in total, we have 6 protons from FADH electrons, 4 protons at complex second site, and 2 protons at complex fourth site. This completes our electron transport system or electron transport chain. Thank you.